Yes, ma'am, we are live.
Good morning to all. It is my immense pleasure to host this seminar series on fermentation technology. On behalf of the Department of Biotechnology, Guru Nanak College, I, Dr. Bharati Ravikrishnan, Assistant Professor and Head Department of Biotechnology, welcome all the
Department of
dignitaries
Biotechnology,
and
Guru Nanak College.
participants to
I,
this
Dr.
august
Bharati
gathering.
Ravikrishnan.
I render my heartfelt thanks to all the participants for their overwhelming response. I wish to have the same participation during all the sessions during the series. As we need to focus on sustainable development, the shift must be from chemical-based product production to bio-based product production. For a large production of products, we depend upon fermentation and bioprocess technologies. This webinar series aims at the basic fermentation technology process and how we can produce products at a large volumes to suit the market demands. Now, I kindly request Dr. Gunita Arun Chando, Vice Principal, Shift to Guru Nanak College, to deliver her felicitation address.
Respected Dr. M.G. Raghunathan, Principal, Guru Nanak College, our eminent guest and resource person for today's webinar, Professor Dobal Mukesh, Professor, Department of Biotechnology, IIT Madras, Vice Principal Student Affairs, Dr. Bharti Ravikrishnan, Head of the Department, BSc Biotechnology, and her team, Deans, heads of the departments, faculty members, dear students, and also participants from educational institutions and industries across India and also from abroad. A very good morning. I deem it a pleasure to welcome Mr. Manjeet Singh Nair, General Secretary and Correspondent of Nanak College, Dr. M.G. Raghunathan, our Principal, the Speaker of the Day, Professor Dobal Mukesh from IIT Madras, Dr. Bharti, Head of the Department, BSc Biotechnology, Dr. Mahendra Kumar, Assistant Professor, Department of Biotechnology, Guru Nanak College, and the host, and the vibrant team of faculty members and all the participants for this national webinar on fermentation technology. This is being organized by the Department of BSc Biotechnology of Guru Nanak College from 13th July to 16th July 2020, and it is comprising of about 7 webinars with eminent resource persons. There has been an overwhelming response for this national webinar. I also welcome on board Dr. Rajkumar from Hubei University, China, Mr. Rajat from Hong Kong, and Mr. Natalija from Macedonia. I also welcome all the participants from Biocon and also from other industries. The topic on fermentation technology is very interesting for sustainable development and also for a positive curricular economy. Product productions must be shifted from chemical-based to bio-based and eco-friendly productions. Fermentation technology is in fact the backbone for bio-based product production. Bio-based products and processes, they cover health supplements, therapeutic, and they also mitigate various environmental issues like bioleaching, biomining, and biodegradation. Fermentation technology has huge potential both in marketing as well as in industry. I'm glad that this topic has been aptly chosen for this webinar series. And this webinar is intended to give a basic yet clear outline about fermentation technologies. I welcome you all once again, and I hope this national webinar series will pave the way for knowledge fermentation technology. Thank you.
Thank you, ma'am. It is now my proud privilege to introduce the speaker of the morning session to you all. Mukesh Dobal, Professor Emeritus, Department of Biotechnology, IIT Madras, Chennai, has agreed to join us and deliver his expertise with us. His expertise are drug discovery, molecular modeling, drug delivery, biomaterial synthesis, bioprocess and bioremediation scale-up technologies, hazard analysis, Six Sigma, SPC quality and design of experiments. His educational qualifications are: he's completed his PhD chemical engineering from University of Aston, UK, postdoctoral research, University of Cambridge, UK, Texas A&M University, USA. He has started his career from 1974 as assistant engineer KCP Limited, Madras. Then, during 1983, he moved to ICI India R&D Center as a group manager, Bombay, and during 2000 as a program engineer in GE Bangalore. And then he started his teaching career from Anna University as an assistant professor in the Center of Biotechnology from 2003. Then moved on to IIT Madras as professor Department of Biotechnology from 2004 till date. He has published 310 technical papers in both national and international journals. He has authored 35 book chapters. He has presented 35 international and Indian conferences. He has published 10 articles in Times of India, Chemical Industry Digest, Chemical Business on R&D management and innovation management. To his credits, he's got 10 patents. To name a few, novel magnesium nanocomposite based ready to use injectable bone cement for bone regeneration, controlled drug delivery, scaffold for tissue engineering. He has also patented a compound transit-mycin effective against bacterial and viral pathogens US 2016. A methods of composition of degrading oil sludge US patent. He has authored 10 books on various subjects like probiotic, bioactive carbohydrates, downstream processing, polymers in marine environment, drug designing, basics and applications, biotreatment of industrial effluent, to name a few. And his books are published by prestigious publishers like Elsevier, Springer, John Wiley, Sons, Marcel New York, Smithers Rapper, UK. The awards bestowed on him are fifth National Award for Technology Innovation in Field of Petrochemicals and Downstream Plastic Processing Industry by Government of India for Innovation in Antimicrobial Food Craft, runner-up 2015. Fellow of Royal Society of Chemistry, England, 2010. Indian Institute of Chemical Engineering, Herdila Award for Excellence in Basic Research in Chemical Engineering, 1995. The Dow Professor M. Sharma Distinguished Visiting Professorship in Chemical Engineering for the year 2012-13 at Institute of Chemical Technology, Mumbai. Professor Kane Visiting Fellowship, UICT Bombay, Lupin India Visiting Fellowship, Dai Iki Karkaria Fellowship, everything is in UICT Bombay, Chinnamal Prize for Best Technical Paper presented in Indian Institute of Chemical Engineering Annual Conference 1994, Commonwealth Scholarship for doing his PhD at UK from 1977 to 1980, and his other credentials are he has served in editorial board of Chemical Engineering, McGraw Hill Publications, USA. His research work on polycarbonate degradation without release of bisphenol A and endocrine disordering chemical has been featured in Scientific American, Chemical Engineering News, American Society, BBC News. Visiting faculty at University of Mumbai Institute of Chemical Technology, a member of American Institute and Indian Institute of Chemical Engineering. He has two startup companies, namely Vital Bioscientific during the year 2013, which involves in design of artificial membrane for studying the fermentation of drugs, and the second company on his credit is T-Venom during the year 2016, where glycan-based shrimp and poultry feeds were synthesized. I immensely welcome you to our webinar on fermentation technology, sir, to share your expertise with us. The platform is all yours, sir. You can begin your talk. Thank you.
Thank you very much for the kind introduction. Let me thank the organizers, Dr. Bharati, Biotechnology Department of Guru Nanak, and also the principal, vice principal of Guru Nanak College. Interestingly, Guru Nanak College and IIT are neighbors, next door neighbors, but we are not able to meet in person because of the current pandemic. So I'm going to talk about fermentation technology. So I am going to cover what are the various products that are based on fermentation. Then I will talk a little bit about the various types of fermenters, a little bit briefly, because there is already going to be a talk on that also. Then I will show you some mathematical modeling of fermentation designs, and what are the issues when you start looking at designing a fermentation process, because you are talking about gas, liquid, solid type of mixing. So this is what my talk is going to be. So if you look at fermentation, fermentation has been there for almost 10,000 years, I would say, from Egyptian civilization, because beverages were fermented for a very, very long time from fruits and other edibles. And India also, it's been there for almost more than 5,000 years. It goes back. And then Asian civilizations have been fermenting a wide variety of food, vegetables, fruits, to enhance their activity, bioactivity, and enhance their stability, and so on. Actually. So fermentation, in some form or the other, has been there for a very, very long time. But recently, if you see in the past 30, 40 years, pharmaceuticals such as antibiotics, hormones, human growth hormones, insulin, antibodies, vaccines, enzymes for diagnostic, industrial enzymes, natural fragrance, flavors, even bulk chemicals like alcohols, acetic acid. So they are all getting being fermented. So the fermenter plays a very, very important role. Uh, in fact, in the entire antibiotics business, you have only through fermentation that's happening actually. So in a fermenter, you generally have a microorganism or sometimes mammalian insect cells. So microorganism could be a bacteria. It could be a genetically modified bacteria. Then it could be yeast, or it could be a fungus. So there could be a carbon source, there could be a nitrogen source, there could be micronutrients for the organism to grow, and with or without oxygen. So some of them are anaerobic in nature, some of them are aerobic in nature. So if you take alcohol fermentation, ideally you would like to not have oxygen into that. If you take butanol fermentation, again, you don't like to have oxygen into that. So some of the fermenters are operated at aerobic, some of them are operated at anaerobic conditions. If you go back in the modern history, the penicillin was the first natural antibiotic, you all know, discovered by Alexander Fleming, that's produced by a mold. Okay, and although it was discovered very, very early, 1928, but large-scale production of this particular antibiotic was carried out in a fermenter much, much later, actually. And then many, many antibiotics, such complicated structures, streptomycin, gentamicin, you can see such complicated structures are produced by bacteria, streptomyces they are called, and they are all manufactured in a fermenter. So if you look at very complex structures, it's very difficult to make it through chemical route or chemical synthesis, but it has to be done only through a bacterial approach. That's a fermentation type of approach. So the bacteria makes use of simple carbon sources and then makes very complex structures. And of course, some modifications are done later on the structure, but the original synthesis of such complex structures are made by the bacteria in a fermenter. So that's where biotech plays a very, very important role. And as you can see now, many drugs are being manufactured through the biotech route. If you look at 15, 20 years back, most of the top selling drugs may be manufactured using chemical approach. But if you look at recent, this is the top 10 selling drugs based on their market value. Actually, there is a mistake here. This should be 14.8 billion. So out of the top 10, except for these three drugs that's marked in red, all of them are manufactured through biotechnology approach, either in a fermenter, followed by a biotransformation, okay, or a biological approach. So as you can see, as things have moved over the past 20 years, the chemical routes are getting replaced by biotech route, which involves a fermenter. So all these molecules, they are large structures, Okay, some of them are peptides, some of them are small proteins. So all these are manufactured in a fermenter using biotechnology approach. So more than 325 million people worldwide have helped by 160 biotech drugs and vaccines. As you know, vaccines are, of course, made through fermentation process. Okay, so it's also being used for developing various kits for testing diseases. Okay, like DNA fingerprinting, HIV test. So here we need quite a lot of enzymes and proteins which are used in diagnostic, and they are all made through the fermentation process. Okay. Human insulin to treat diabetes. Okay, that comes out through a fermentation process. I will show you the flow sheet of that after some time. So that's again is based on fermentation because we need to have large amount of insulin. So that's produced in a fermenter. So if you look at just for data, top 10 pharmaceutical companies, top 10 biopharmaceutical companies. So the original pharmaceutical companies were working on chemical approach, chemical synthesis, chemical transformation for manufacturing drugs, and they have slowly moved into biotech approach, which involves the fermenter as heart of the manufacture, followed by purifications downstream, and then some biotransformation and so on. So all these pharmaceutical companies which were working on chemical synthetic approach have moved into biochemical based approaches. So a fermenter is generally the heart of the entire manufacturing process. So on the left hand side you may have the upstream. I think you are going to have a talk on upstream, and the right hand side is the downstream. Okay, again, I think you are going to have a talk on downstream. So the fermenter is the heart of the entire bioprocess here. So the fermenter may be making your product. It could be a vaccine, or it could be a protein, or it could be a drug, biological drug at certain purity, which gets purified in the downstream to a high level or high degree. So the fermenter is the heart of the entire um, biological process, actually. So there are two things that can happen, either the fermentation or biotransformation. Okay, there is a slight difference. There is a difference, not slight, a lot of difference between the both fermentation and biotransformation. In biotransformation, you have a reactant which is converted to a product. So you may have oxidation, you may have reduction. So there could be an oxidoreductase type of enzyme which may be putting in some oxygen, or it could be putting in OH groups, or it may be removing the H groups. So the enzyme or bacterial may be doing that job. So that's called biotransformation. Whereas in fermentation, uh, very simple sugars um, may be getting converted into very complex products. Okay, that's fermentation. The biotransformation is converting the reactant into product, either by oxidation or reduction. Okay, whereas in fermentation, sugars, different types of sugars are converted into very complex products. Okay, to get your vaccines, or to get your antibiotics, or to get your proteins and so on. Actually. Okay, so some of the products are produced intracellularly, and some of them are produced extracellularly. So you need to recover the intracellular material um, by disrupting the cells and then going into various purification processes, product extraction, product purification, and so on. I'm not going to spend a lot of time on this because that's not the focus of this talk, actually. In fermentation, you can have intracellular products, you can have extracellular products. Some products are producing Golgi bodies, and some products are produced in the periplasmic region. Okay, and so you may have to recover the product. That means you have to break the cells and recover your product. Some of the products are produced extracellularly, which is then your things are very simple. all you have to do is collect the supernatant and recover your product. Whereas if it is produced intracellularly, we need to collect the cells, break the cells, and then break the cells and then get your product out. So that's extra steps that are involved in the product recovery and product purification. Okay. So there is something called low value, high volume product and high value, low volume product. So if you look at ethanol, antibiotics, they are quite cheap. Per liter, ethanol may be costing 20, 25 rupees a liter. Antibiotics may be costing few rupees a gram and so on. So they are all low value product, but the profit comes from the volume of the product. Okay, that's called low value, high volume product. That means you produce large quantities, thousands of liters of ethanol, but the cost of the ethanol per liter is quite cheap. Okay? So you have to reduce the operating cost in this. A lot of strain improvements are done. As you know, ethanol fermentation over the past 30, 40 years, there is a lot of changes in the strain development, and the strain, current strain is able to tolerate large concentrations of ethanol. Okay? So strain development is a very important step in low value, high volume product. That means engineering the strains. You need to reduce overhead cost. We can think about convert batch into continuous process. Economy of scale. That means when you increase the volume, the capital cost comes down, and so the profit increases. Okay, so this is what the strategy followed for products like this. It's a low value, high volume product. Whereas if you look at high value statins, okay, statins, as you know, there are many statins are used for thinning the blood. Those who have cholesterol related issues take these type of statins to thin the blood. Okay, statins are called high value product. So a gram may be costing thousands of rupees when compared to say an antibiotics. A gram may be costing few rupees. Insulin. So all these are called high value, low volume product. Okay? So um, even if you make small quantities, because the cost is so high, you make a profit. Okay, so product recovery is very important here. So the downstream becomes very important in this high values situations, because even if you lose small amount of your product, you are losing a lot of money. Purity is also important because if you're talking about biopharmaceutical, the purity plays a very important role. And of course, if the competition increases, the high value becomes low value. If there are more industries which start making statins, then the cost of may start coming down, and it will become from high value to low value. So depending upon whether it's a low value or a high value, the fermentation situations change. The downstream also changes. Okay? So in high value, the product recovery is very important. Whereas in low value, strain improvements, process optimization, those become important. Even if you lose little bit material in the downstream, it doesn't matter because it's low value. So strain improvement is very, very important here. Process optimization, improving the process conditions to increase the yield. Those factors become very important if you're talking about low value product. Okay. So after the fermentation, we have many downstream steps for the separation of your product and purification of the product. Actually. So generally you will have the biomass or cells that needs to be removed. As soon as the fermentation is over, you are going to have dead cells, cell debris. You are going to have salts because we add many micronutrients during the fermentation process. Okay, then so that all those are removed either through filtration or centrifugation, some solid liquid separation process. If it's an intracellular material, we have disintegration of cells. Cells needs to be broken down, either mechanical method like ball mill, or it can be a homogenizer or a bead mill. And then separation, you may be using some sort of a liquid extraction. You may be using salt precipitation, and then final purification. You may be using different types of chromatography to get the purity. Depending upon if it's an animal feed or human, the purity levels may change. So you may have different types of chromatography to purify, and finally drying with a solid, and then stabilizing, because your product may disintegrate or may not be stable at higher temperatures, or it may catch bacterial infection. So you may have to add some stabilizers, some antioxidants, and so on actually. So these are the downstream steps and that are done after a fermentation. So I talked about, yeah. So let's go back to various products that are coming out through fermentation. The first one is, of course, the penicillin. Although the penicillin was discovered very, very early, okay, again, it came back only much later, okay, because the manufacturing of penicillin, uh, large-scale production of penicillin was a big issue. That means large-scale production using a fermenter. Actually. So it became very important because of Second World War, when the soldiers were dying because of infection. They needed an antibiotic. So there was a need, very dire need for an antibiotic. So some of the technologies were developed to produce large-scale penicillin in a large fermenter, as well as in purification. So penicillin produced by an aerobic submerged fermentation actually. If you look at it in 1945, it was 1 milligram per decimeter cube using an organism called penicillium. Whereas in 2015, again, Penicillium chrysogenum, there is a different species, and the yield has improved by almost huge amount of time. Okay, 50,000. Okay, so that's a very, very important point we need to keep in mind for low value product. Continuously, we need to improve the yield. Process optimization can help only up to certain level, but beyond that, we need to think about genetically modifying the organism so that it keeps improving the yield. Okay, so as you can see, in this period, the yield has gone up tremendously. So the cost of the product also has come down. So the quantity of production also has increased tremendously. If you look at antibiotics, they can be selectively toxic by targeting bacterial cell wall, 70S ribosomes, and enzymes that are specific to bacteria. Actually, so generally, human are not affected. So after penicillin, other antibiotics in that same family started getting discovered: ampicillin, amoxicillin, methicillin, a lot of which inhibit enzymes involved in synthesis of peptidoglycan of the cell wall. Okay, they are called bactericidal. Okay, in you have the bacteriostatic and the bactericidal, two different approaches by which antibiotics and antibacterial work, and this is called bactericidal. Okay, so they are generally for gram-negative bacteria. There are many other types of antibiotics affecting cell membrane, DNA replication, transcription, translation, and so on. Actually, okay. So if you look at penicillin production, the fungus Penicillium chrysogenum, you have sugars, lactose and other sugars. Then you have the nitrogen source, yeast extract. Okay, all the antibiotics like penicillin is a secondary metabolite. That means the product is formed not during the growth phase of the bacteria or the organism, okay, but at the stationary phase. Okay, and it's produced by a fed batch process, because when you have a fed batch process, you are adding the carbon so slowly, so you are prolonging the stationary period, so the production also improves or increases. So typically, if you look at penicillin, excuse me, yeah, if you look at penicillin production, the lactose gets consumed. The x-axis is your number of days. The lactose gets consumed. and biomass increases directly proportional to the lactose consumption, but you don't get your product, and you start getting only after the third day. Okay? That's why it's happening at the stationary phase. So it's not called growth associated, but it is called non-growth associated because they are secondary metabolites. If it's a primary metabolite, you will start having production with the carbon consumption. Okay? So because it's a secondary metabolite, you have the product production much later when compared to the carbon consumption actually. So many of the antibiotics you will see this type of trend. The product formation happens much later, almost closer to the stationary phase. Okay? The first few days you will not see the product formation. So penicillin is, like I said, extracellular product. So when you have an extracellular product, it becomes very easy to purify it. Whereas intracellular, we need to collect the biomass, and then we need to break the cells to recover your product. So extracellular, we just remove the biomass, and then product needs to be purified. So it is dissolved and then precipitated as a potassium salt. As you know, precipitation always improves products. Precipitation followed by crystallization always improves the product purity. And also potassium, most of the drugs, if you see, are salt form because it improves the aqueous solubility in the GI tract. Okay, so typically this is what happens. A typical process for penicillin manufacture, we have the batch fermenter. Okay, 6 days. Then you have a filter. The fungal cells are removed. Then we take the filtrate. Then butyl acetate, and then we make a potassium ions added to it, and then you get salt, and then you filter it again, and then you purify it. So it's become a potassium salt. So this is quite a simple downstream because it's an extracellular product, but the product has to be made into a salt. But it's a very long fermentation process because we are talking in terms of days, almost 10 days. So this will be the norm for most of the antibiotics. You'll be running your process for several days. Okay. And then, of course, you have different types of semi-synthetic penicillins, as we call penicillin B, penicillin O. All these are biotransformed from the original penicillin. Okay, so they are enzymatically modified to make a variety of penicillins with slightly different properties. So you have a fermenter which makes your penicillin, and then you have a bioreactor which biotransforms into different types of penicillin. They are called semi-synthetic penicillin, which have different properties for different type of applications and so on. Actually, okay. So the flow sheet, if you look, this is how the flow sheet looks. Here is your fermenter, and here we have the downstream. Of course, you are acidifying the penicillin, then neutralizing, then you are crystallizing the penicillin into potassium salt. Then it is, it is purified. I mean, it is filtered here and then dried using a fluidized bed dryer and then stored. So this is the entire downstream where you are making potassium salt of the penicillin that's coming out here actually. So here you are removing the biomass by filtration because it's an extracellular product. The supernatant is what is desired for you. So let me show some flow sheets for various products that are manufactured using fermentation route. Of course, ethanol, Acromyces cerevisiae, okay, this is used for the production of ethanol. Different types of sugars are used, exos. Okay, so 70% of the cost is raw material cost. Cane sugar used in Brazil and India, whereas USA uses corn sugar actually. And the brewing produces dilute concentration of ethanol because one of the big problems in ethanol fermentation is the organism is not tolerant to high concentrations of ethanol. Okay, although there is a lot of genetic modifications that are being done to improve the alcohol tolerance of organism, you may get 15 to 20% tolerance strain. So we need to collect the dilute products and then do distillations to recover your product actually. So this is a thermophilic organism, 50 to 60 degrees centigrade. Okay, it's an anaerobic process actually, and then cycle lasts for 30 to 40 hours. So this is a typical fermentation approach used. Okay, so after the fermentation, um, we need to concentrate our product. The cells have to be removed. Okay, then the product is concentrated using distillation. And this is the flow sheet for ethanol production. You can see the fermenter here, and then we have the filtration taking place, distillation. So it's a quite simple downstream also, because ethanol can be easily distilled. So we can get a different purity of ethanol by having more distillation columns, and so on. So the solid biomass is used for dried and can go into animal feed. Okay, this is where you have the fermenter. Generally, these fermenters are big wooden cask slide, or it can be even a mild steel type of facility. So large fermenters are used. And as I said, for low value, high volume products, the size of the fermenter plays an important role. The larger the size, over is lower is the overall capital cost contribution in the final product. So a lot of companies manufacturing ethanol, as you can see, millions of gallons per year. Some of these numbers you can see millions of gallons per year. Okay. Then comes amino acids. This is also a very big business, through fermentation and biotransformation, because a lot of amino acids are used as animal feed additives. Amino acids are used as flavor enhancers, like monosodium glutamate. So we all have come across all the food flavor enhancers, meat tenderizers that uses monosodium glutamate. Aspartic acid, this is the artificial sweetener. Then amino acids are used in medical field, and these amino acids are used quite a lot in animal feed additives, okay, and so on. So glutamic acid, lysine, they're all made by fermentation, and methionine is made through chemical route, and aspartic acid, alanine, again through enzymatic synthesis of one or two steps. Okay, so amino acids is a very big business where fermentation plays a very important role here. So major producers of amino acids: Japan, US, South Korea, China, Europe. Okay, so it's a very big business for monosodium glutamate, aspartic, phenylalanine, and so on. So they are used in dietary supplements. Okay, like I said, animal feeds, medical feeds, food and flavor enhancers, and so on. Actually, and then of course, amino acids they have to be coated to prevent its degradation and improve its shelf life and stability of the amino acids. So if you look at the cost contribution, 67% of the cost is due to the nutrient, and 12% is ammonium hydroxide is used in ion exchange column chromatography, and then 13% is the cost of the coating, and that's done on the amino acid product to improve the shelf life. Um, if you look at the equipment cost, 33% of the cost is because of the reactors. The remaining is all downstream. 33% of the cost is the fermenter cost. As you can see here, that plays a very important role, the fermenter cost in the total equipment cost or fixed cost. So a lot of industries are involved in amino acids production. We'll not go into that. Reactors, almost 33%. The rest of it is all downstream, the manufacture of leucine and lysine manufacturing. This contribution is towards the fixed cost. And similarly, if you look at Other aspartic acid and phenylalanine, again 31% of the cost is the reactors, and remaining is all the downstream equipments like heat exchangers, ion exchange column, filters, and crystallizers, and so on. So the fermenter cost plays a very important role in the total fixed cost. Human insulin is another biotech product which is made through fermentation. As you know, insulin is extremely important, has become very important because of the diabetes. Okay, so here again, reactors and hardware related to fermentation takes 62% of the equipment cost. So the fixed cost contribution by the fermenter is almost 60, and the remaining 62, the remaining 30% goes to the downstream processing equipment, which may be filtrations and centrifugations and chromatography and purification and so on actually. Okay, so as a typical flow sheet of a recombinant insulin, so here we have the fermenter, then we have the cell recovery. Here the product is inside the cells as a Golgi body. So we need to solubilize and get the product out of it actually. That is why we have the cell disruption. I will not go into the purification here, but as you can see, this is the fermenter, or call it the bioreactor here, and this is where you are recovering the intracellular material, and then you are purifying it using different types of chromatography to arrive at highly pure active insulin. Because it's a human use, the purity levels have to be very, very high. We'll not do that. Okay, again, you can see the reactions. The fermenters take about 50% of the equipment cost. Acetic acid or vinegar is again manufactured using fermentation. Okay, vinegar, as you know, is used quite a lot in soft drinks, cellulose acetate, photographic film, PV, polyvinyl acetate, wood glue, synthetic fibers, fabric. So it has got a very big business, and it's produced both by synthetic or bacterial approach, and the biological, that is the fermentation, takes care of only 10% of the whole production, and that is called the vinegar, which is generally used in food and flavors, whereas the industrial applications or product is made through chemical route. That's by methanol carbonylation. Okay, so acetic acid is produced by these acetobacter. This is an aerobic process actually. Even Clostridium also produces some of the acetic acid, converts sugars to acetic acid. So there are many organisms which can make. Yeah, this is the flow sheet. As you can see, we have the fermenter here, and we have a solvent extraction to remove the acetic acid, and then there is a distillation column which removes your solvent and which recovers your acetic acid. So this is a typical flow sheet for acetic acid production using a fermentation approach. Then you have 2,3-butanediol. This is a very valuable fuel additive. This is used in the manufacturing of printing inks, perfumes, fumigants, softening agents, explosives. This is produced by Bacillus and Klebsiella pneumonia, two different organisms. They use quite a lot of sugars, hexose, pentose, disaccharides, and so on actually. Okay, so that's 2,3-butanediol. And of course, the ethanol is one of the side products of this. And then butanediol itself can be an inhibitor. So that could be one of the problems in fermentation. When you're doing a batch type of fermentation, when you're adding large amount of raw material, that could be an inhibitor for the organism. So you may have to resort to fed batch approach. If the product is going to be inhibiting in a fermentation, then you should think of a strategy of continuously removing the product so that you don't increase the amount of product in the fermenter and thereby stop the reaction. The example there is butanol production. So we have Clostridium, which produces butanol, but butyric acid is one of the side products which may be toxic to the organism. So if we can think of removing butyric acid continuously, then you may improve the life of the organism to produce more. Okay, butanol. So we have sometimes substrate inhibition happening in fermenters, where you need to use a fed batch type of approach, or you may be having a product inhibition type of situation happening, where you must think of continuously removing your product or side product continuously, so that you don't let it accumulate. So these are some points we need to keep in mind when we are thinking about handling a fermenter with these type of situations. So butane diol, as you can see, a fermenter, then we have filtration, then we have solvent extraction, and then solvent is being distilled out, and then your butane diol is recovered here. So this is a typical flow sheet for butane diol production. This is another type of flow sheet. We will not go too much into the downstream, but again, the main is your fermenter. Okay. There are many companies which are involved in the manufacture of butane diol. European companies and US and Chinese companies here. If you look at monoclonal recombinant antibody, again, you have a fermenter and then the product purification, because it's a product consumed. It's meant for human applications. We have several chromatographies coming into affinity chromatography, ion exchange chromatography, hydrophobic interaction chromatography, and so on. Actually, so a lot of chromatographies. Then if you look at recombinant human antitrypsin, again, we have a large number of downstream. This is an intracellular product. So we have a cell disruption that is taking place here. And the fermentation is your heart. Streptomycin, another antibiotics. This is produced by a particular organism. Okay. So again, this is made through fermentation. Okay, a lot of. So we looked at several products, um, small value products like ethanol, biopharmaceutical products like insulin or antibodies, all made through fermentation. Some of them are an intracellular product, some of them are extracellular products. So the strategies which the fermenter has to work on changes depending upon whether it's an intracellular, extracellular, whether it is a low value product or a high value product. I will show you some pictures of different types of fermenters. I will not go too much into that. There are a wide range of fermenter designs available when you decide to go on. There are a wide range. The could be a solid state type of fermenter. It could be a slurry fermenter. That means you may have biomass, you may have air bubbling through and a liquid. It could be a gas-based fermenter or it could be a liquid-based fermenter. Okay, and then the mode of operation could be a batch process, fed batch process. It could be an aerobic process. It could be an anaerobic process. So different type of thoughts we need to give in when we are designing a fermenter. So solid state fermentation has been there also for a very long time historically. What you do is you deposit a solid culture substrate such as rice or wheat bran on flat beds. Then you seed with the microorganisms. So the whole fermentation takes place in a solid form. It's not very uniform because it's going to be heterogeneous, unlike a liquid state fermentation or a submerged fermentation. In a solid state fermentation, because it's a big solid mat, the fermentation might not be uniform, the growth of the organism might not be uniform, but it is good. It's very, very cheap, and it's used quite a lot for fermenting food products, bread, cheese, and so on. Actually, so generally these run for several days. It's a temperature control. So all you have to do is you have a big flat bed of your medium, and you have a big, and then add your microorganisms, and then allow it to do the fermentation. That's called a solid state fermentation. But process control is very difficult. Uniformity of the process is very difficult. That's why solid state fermentation is not used if you want to have high value products. So a typical fermenter, this is how a typical fermenter looks like. We have in agitator. Of course, there are non-agitated fermenters are also there. I will show you some pictures. So we have an agitator here, and then we have the liquid here. A gas may be bubbled in for aerobic, or if it's anaerobic, you may maintain either facultative anaerobic, you may maintain nitrogen, or you may maintain carbon dioxide, fully anaerobic, and so on. Actually, so in a batch fermenter, we feed everything inside and do the reactions for many days, and then we discharge. In a fed batch, we start with small amount and we keep on adding the feed, slowly build up. Okay, that's a fed batch. In a continuous, you have a continuous feed in and we have a continuous product that's going out. And of course, you are going to have gases coming out, carbon dioxide, carbon monoxide, maybe methane, maybe if it's an anaerobic fermenter, and so on. So this is a typical submerged fermenter or liquid phase fermenter. There's an agitated fermenter. You can have non-agitated fermenter also. Let's not go into that. These are different modifications to the agitated fermenters. You may have a draft tube. The idea is to improve the mixing part. Okay, the idea is to improve the mixing part by having a draft tube. So the gas liquid moves up and down because of the presence of the draft tube, and the agitator. Okay, because when we talk about large volumes, mixing of gas liquid is a big challenge, and maintaining the biomass suspended is also another challenge. Then we have multi-compartment type of fermenters here. As you can see, it's tall fermenters with the many compartments. So each compartment may be acting as a small continuous stir tank fermenter. So it's almost like multiple bioreactors stacked one on top of another. This is called a jet loop reactor fermenter. As you can see here, there is no mechanical agitation. The gases is coming in here, and you recycle or you achieve mixing by having an external pump which puts the product, circulates the product up and down from this. Okay, then we have a T plate type of design. Again, we have multiple plates here to bring in the mixing and breaking of eddies and so on. Actually, this is called airlift loop reactor. These have become very popular nowadays, because mechanical agitated systems, beyond a certain volume, it may be very uneconomical or unwieldy to have very large mechanical agitators. So these type of airlift fermenters have become very popular, which can handle large quantities of the broth. So here, as you can see, there are no mechanical agitation. So we have long tubes here. So the gas liquid rises because of buoyancy, and the gas gets disengaged and the liquid comes down. So you create here up and down movement of the liquid with the help of the gas flowing in from the bottom, and by having a large tubes. These are called airlift loop bioreactors. Like I said, these are used for large fermentation processes. Mechanical agitated vessels can be used only for medium to small, because it becomes very unwieldy and economical to have designed very large agitator motors. So airlift loop fermenters have become very popular. Okay, then you also have bubble column fermenters. Here it's almost like airlift, but you do not have the draft tube here. Very large, very tall columns. So because of the height of the column and the hydrodynamic height, the gas pressures are, I mean, the pressure here is very high. So the gas gets dissolved and solubilizes into the liquid. So we achieve good gas liquid mass transfer in these type of bubble columns. So if one wants to go for non-mechanical agitated fermenters, you may go for airlift or bubble column. Another advantage of airlift or bubble column is, especially if you are handling very sensitive cells which are very fragile, like the animal cells, which have very thin cell walls, which may break because of mechanical agitation. Then we cannot use mechanical agitated type of fermenters. We may have to go for airlift bioreactors or bubble column bioreactors. So we have many types of designs. These type of designs are used for immobilized um, uh, bacteria or immobilized enzymes. So many types of designs. I will not go into that because of short of time. These are called fluidized bed fermenters, where you immobilize your enzyme or immobilize your bacteria on very particles which have low bulk density, so they are able to float. Thereby, you achieve good suspension of the biomass, and thereby you get a good mass transfer things. So when you are designing a reactor, we need to consider the kinetics of reaction, including inhibition. Like I said, it could be substrate inhibition, product inhibition. Then we need to consider mass transfer from gas to liquid. If it's an aerobic, we are bubbling in air, so air has to dissolve into the liquid. Then mass transfer from liquid to surface of the microorganism. So the organisms should have enough oxygen for growth. So the oxygen uptake rate, OUR as we call it, should be comparable to OTR, oxygen transfer rate. If you are not going to have enough transfer of oxygen, we are going to have anaerobic situations, and the microorganisms might not grow. Heat transfer, as you know, fermenters, we cannot increase the temperature too much. Organisms may lose their activity, so we need to consider how to remove heat that's liberated. Mixing and agitation. There has to be a uniform mixing inside the fermenter. Type of fermenters. I just quickly showed you many types of fermenters. So which fermenter do I select? Based on the product which I am producing, based on the microorganism or cells which I am using. Physical properties of the medium, viscosity, density, surface tension. Physical properties of the microorganism. Is it a thin-walled or a thick-walled, or it's a filamentous type of microorganism, or it's a mold-like microorganism. So the physical property. And the mode of operation. Should we go for batch? Should we go for semi-batch or a fed batch? Should we go for a continuous reactor? So different points we need to keep in mind when we are designing a fermenter. Unlike a simple chemical reactor, because in a chemical reactor we are mixing and reacting reactants either with or without a metallic or a non-metallic catalyst. So here we are dealing with microorganisms like bacteria, fungus, yeast, animal cells, which are live, which grow. So we need them to be growing in optimum conditions, actually. The cell growth process, as you know, in a fermenter, you can have the lag phase, then we have the log phase and stationary phase. So there are different approaches based on which various processes have been categorized. The fermentation. This is called a Gadden's classification, especially for batch fermentation. The type 1, the product is derived directly from the primary metabolism. Okay, okay, that means we have here growth associated process. Okay, that means if you have the product produced as a function of time, it will be directly proportional to the substrate consumption. So that's called a growth associated. Growth associated. That's type 1 type of process. Whereas in type 2 process, there could be two maxima in the batch fermentation. Okay. So initially you may have high substrate consumption, but there is no product formation. Growth slows down, and then we have product formation again with a high substrate consumption. So you have citric acid, itaconic acid, amino acids. You will see two rates for the substrate consumption. That's the called type 2. If you go for type 3, okay, the product is not derived from catabolism, but from amphibolic pathways. So both product formation and substrate consumption appear to be very, very different, actually. Okay, so it could be because of secondary metabolites, many antibiotics. Like I showed penicillin, where it's happening at this stationary phase. The raw material gets consumed fully and then finally. So you may have possibly negative specific yields. D biomass by dt divided by 1 by biomass less than 0. So these are three types of processes as classified by Cadden. There is another classification that's called the Deindorfer's classification. Okay, so your primary substrate getting converted to product, or primary substrate getting converted to product and some other side product, primary substrate getting converted to some intermediate, finally product, then nutrients getting converted to intermediates before conversion to product. This okay. Um, this is four types. Okay, this is called stepwise, this is called consecutive, this is called simultaneous, this is called simple type of process actually. So this is called Deindorfer's classification. So we have the simple, okay, A gets consumed, product is formed. Simultaneous, A gets consumed, you are getting product, and something else like cell mass is formed and fats are formed. Consecutive, you get A gets consumed to B gets consumed to product. Okay, this is another type of situation. And the fourth type is we call it the dioxic growth or stepwise. No, E. coli growth in glucose. Okay, it consumes glucose fully. Once glucose is consumed, then it may take sorbitol, another substrate, and go into your product. Okay, so we have four types of situations that may be happening. Okay, in Deindorfer's classification in the product formation. Okay, so scale up is also very important point we need to keep in mind when you are looking at fermentation. As you know, we do experiments in lab, maybe in rotary shakers, flasks, in 100 ml quantity, but finally we may be manufacturing the product in 100,000 liter capacity. We are talking about almost 1 million times scale up. So because we are handling microorganism, it should be working at the same conditions as it was in small scale when it goes into a large scale. So scale up, there are a lot of scale up equations available by which one can think of scaling up from very small scale to large scale. Okay, we'll not talk about it actually. And there are a lot of mathematical models also available by which we can think of modeling the fermentation process actually. Okay, we call it batch cell growth, we can call it continuous cell growth. We can use these mathematical models for identifying doubling time of the microorganisms. We can use it for predicting the volume of a batch culture because of want of time will not go into. Whereas if you look at molds and filamentous organism, they have a different set of equation where this is called the mass of the mold. It changes as a function of T cube, time of cube. Whereas when you look at bacteria, they double and multiply and grow, whereas molds grow in the amount. Okay? So will not go away. So there are many equations, like I said, if you are designing a batch reactor, fed batch reactor, extended fed batch, continuous stir tank. So based on the kinetics of reaction, we can calculate the batch cycle time, we can design the volume of the fermenter, and so on. Again, because of want of time, I will not go too much into this different modeling approaches by which. And then if you are talking about a monod chemostat, how to go about designing the volume of a monod chemostat, and the flow rate that is required to achieve maximum growth. And so, of course, there are a lot of non-idealities in a fermenter. Large fermenter with mechanical agitation, we can have dead zones because of improper mixing, as you can see. And even different types of blades can lead to different types of mixing zones, improper gas-liquid contact, so low rate of reaction. Okay, then we are talking about mass transfer. We have the pellets, and that is your microorganisms forming flocks or pellets. Then we have the gas coming in, then gas film, then gas dissolves in the liquid, reaches your solid flocks or solid biomass. So many resistance are there. So we need to keep those points in mind when we are designing gas, liquid, solid mass transfer processes actually. So we can bring in mass transfer limitations when we do a modeling of that. So mass transfer in the gas phase, mass transfer in the liquid side, and so on actually. So if you want to design oxygen transfer rate based on the oxygen uptake rate, we'll not go into that. And of course, if you are talking about enzymes, immobilized enzymes which are immobilized on solid surfaces, the substrate has to reach the enzyme. That means the enzyme might be inside the pores of a solid surface. So we have the diffusion of the substrate into the solid support. So there are equations for designing those diffusion situations as well, especially for solid supported enzymatic processes. Okay, let me conclude what I wanted to talk about. Fermentation has become very important in biopharmaceutical and of course also in pharmaceutical industries. As I showed you, most of the top selling drugs are now made through fermentation route. So drugs, antibiotics, nutraceuticals, industrial enzymes, enzymes used in diagnostics, um, um, bulk chemicals like your acetic acid, like your butanol, ethanol, all these are now being made through fermentation routes. So, because unlike a chemical reactor, fermenters contain microorganisms or fungi or yeast or cells which are live, which needs growth, proper carbon, nitrogen, micronutrient, oxygen, pH conditions. We need to design the reactor properly to take care of all these issues. There are different types of reactors one can think about. I showed you how to design reactors based on the type of process you are talking about, the cost. Then comes the scale up, because as you move from small scale, 100 milliliter scale to 100,000 liter scale, and then the downstream, that's not the focus of my talk, but downstream also plays a very important role. When you want to recover your product, when you want to purify your product, downstream plays a very important role. So fermenters have become very, very important in the manufacture of biopharmaceutical products, as well as animal feeds, as well as nutraceuticals. Thank you very much for your time. Let me thank the organizers of Guru Nanak College for giving me an opportunity. Thank you very much.
Thank you, sir. It is a great indeed a pleasure to listen to your enlightened talk, sir. You have really enlightened us with your knowledge, and you have shared your expertise. And there are a lot of positive feedbacks even in our YouTube chat box. Now, I request Dr. Mahindra Kumar, Assistant Professor, Department of Biotechnology, to take over the discussion session.
Thank you very much, ma'am. Sir, shall I post a few questions which have been asked by the participants so that you can answer, sir?
Yeah, sure.
Yes, sir.
Yeah, there is a question about bio-based product at home. So if you are talking, of course, if you are talking about products like wine and alcohol and food, they are all a home industry, and they are made using home appliances only. But if you are talking about pharmaceutical products or vaccines, then of course you can't do that at home, because we have certain GLP based situations, conditions which we have to think about. But of course, like I said, bio based fermentation of sugars and vegetables and fruits for producing wine and alcohol are being done at home actually. So there are nowadays a lot of kits available also, which has these organisms in the lyophilized state, and one needs to just follow that, and people do make wines in the western countries. I don't think that's allowed in India, but I think in west they do that actually. Okay, so somebody has asked some light on fermenters used for animal cell. Okay, so as I mentioned, animal cells are very fragile. They have a very thin cell walls. So generally, you cannot use agitated type of fermenters. You need to go for different types of fermenters which does not use agitation at all. I showed you a couple of pictures. Okay, where they do not airlift type of bioreactors or fermenters where there is no agitator. So we need to keep that in mind because the major problem for handling animal cell is they are very fragile, so they break down. Uh, and also they are more sensitive than bacteria or yeast. Or even fungi. Yeast and bacteria are a little bit more can take up harsh conditions, but animal cells are very sensitive with respect to some of the pH and temperature conditions. So the control of pH, temperature, non-use of agitation, or design of a bioreactor accordingly is a main point which they keep in mind when they are designing animal cell-based fermenters. Dioxic growth related dual fermentation. So dioxic growth happens when we have two different raw material, and when you have two different raw materials. Okay, so the organism makes use of one raw material completely, and once it has used that raw material, it goes and starts using the other raw material. Okay, so generally dioxic growth happens in such a type of situations. So you need to think of whether you really need to have that sort of situations where we have two raw materials added. So we can always think about adding one raw material after the other raw material. But when you are going to add another raw material, you are going to have situations like um, again lag phase that's happening because the organism have to get used to the new raw material also. So you may end up having two lag phases, the original lag phase as well as the second lag phase. Okay. Biotechnology student, Satya Mohammed.
No, 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 sir. One student has asked. um,
Okay, any other question?
yes, sir. Yes, sir. I'm just going through the session, sir.
Yeah. Be suitable for small scale production of commercial enzymes. Commercial enzymes are very harsh. Though, like commercial enzymes, when you talk about washing, okay, detergent manufacture. So generally, agitated type of fermenters are good enough for commercial enzymes. So it could be a batch type of agitated fermenters for commercial. So advantage of commercial enzymes are they can withstand very harsh environment. So they are easy. Of course, commercial enzymes, you are talking about cheap products because the costs are very low. So we need to go for large scale manufacture. So generally, scale is a very important aspect. So by having a large scale, you achieve the contribution of the fixed cost into your final selling product. Does organism has to be modified for which process? No. um, See, nowadays, if you look at many fermentation processes are done using genetically modified organism only because 2 points. Number one, the wild strain has certain yield, but we can keep improving the yield using genetic methods. So you can keep increasing the yield because that improves your selling price or even the cost of reduces the cost of manufacture. Number one. Number two, wild strain may keep changing their conditions with time. So if you want to work for the next 10 years, 15 years manufacturing a product, it's always good to have genetically modified strains. So genetic modification has become part of fermentation technology. In fermentation, the students are required to know many things when joining jobs. Yeah, so fermentation is not just chemical engineering where the person will learn only bioreactor, agitation, temperature, pH. But like I said, genetic engineering also has become part of fermentation because as time keeps going, one needs to keep improving the strain conditions either improving the yield of the strain or improving its operability and so on. So genetics also has become part of fermentation. Then of course you need to know about upstream processing, downstream processing. So a typical student should know about when you talk about fermentation technology, should know about fermenter design, upstream processes, that means industrial microbiology, downstream processing, that means purification, centrifugation, chromatography, as well as genetic engineering. So all these are part of has become part of fermentation technology, which fermenter is used to extract secondary metabolites from plants in large amounts. So secondary metabolites means they are not growth associated. They are generally non-growth associated. That means you will start getting the product only after the substrate is consumed. You end up reaching the stationary phase. So even normal fermenters can be used, but you will start getting the product only slightly later, unlike a situation where you are more in where you have primary metabolism as your desired product. So normal fermenter, agitated fermenters can also be used. Can you produce bioethanol from materials like fruit waste? Yes. Like I said, ethanol, all alcohol production has been practiced even at home, using different types of fruits, vegetables, and so on, using different organisms. So it can be done also, actually. Yes, using Saccharomyces type of strains, ethanol is being produced at home also. But the only thing is, I don't know whether it is legal in India to produce ethanol at home. So one needs to keep that in mind.
Okay, sir. That's all. We have a lot of appreciation and thanks message to you, sir, from the
Thank you very much.
participants.
Thanks. Thanks. It's a pleasure.
You.
Once again, thank the organizers and especially Dr. Bharati also for inviting me to give a talk. Thank you very much.
Thank you, sir.
Thank you, sir.
Thank you.
I request all the participants to just wait for another 5 more minutes that we'll be posting the feedback link for you. And now it is my great privilege to call upon our Vice Principal Ma'am, Dr. Gunita Arunchando, to give her concluding remarks.
Okay.
Ma'am, please unmute your mic, ma'am.
Ma'am, kindly unmute your mic, ma'am.
Yeah, I have. Yeah, it was a wonderful session by Dr. Mukesh from IIT. He shared his views with all the participants, and I could see that there were a number of questions also. And I thank you so much, sir, for
Thank you.
spending your time and sharing your expertise both with our students and with all our participants, because our participants have included research scholars, faculty members, people from industry, and our students also. So thank you so much.
Thank you.
Thank you,
Thank you.
sir.
Thank you.
Yeah,
Okay.
and I thank I thank Dr. Bharati Ravikrishnan also for organizing, for taking this initiative of organizing this wonderful series, webinar series throughout the next 3 days. And I think she has about 7 webinars. So I wish you all the best, and I'm sure you will bring in just the way we had Dr. Mukesh with us, a very eminent resource person. I'm sure all the other resource persons are also very eminent. Good initiative taken by Dr. Bharti and the Department of Biotechnology. And I also thank Dr. Mahindra Kumar for being the host and also for coordinating the events in the department. Thank you.
Thank you, ma'am. To all the participants, the feedback link will be posted in your registered mail IDs. So anyone, if you want to give your mail ID, you can put it on the chat also. We will send the feedback link to your registered mail ID. You can fill in the feedback forms. Now, it is my pleasure to render my gratitude, my heartfelt thanks to our honorable speaker, Professor Dobal Mukesh, to share his expertise and patiently clarifying the doubts. Respected Mr. Manjit Singh Nair, our General Secretary and correspondent, who has been supporting us in all our endeavors in his absentia today. Dr. M.J. Raghunathan, our principal, both vice principals of Shift 1 and Shift 2, my dear faculty fraternity of Guru Nanak College, students, and to all the most valued invited guests, ladies and gentlemen. My special thanks to my students, Balaji, Manikandan, Dakshita, Susan, and to all my department colleagues, Dr. P. Mahalakshmi, Amalraj sir, Dr. Mahendran, for the support in bringing up this webinar series in a successful way. Thank you all. Over to now to Dr. Mahendra Kumar for few more instructions.
Thank you, ma'am. Dear participants, we thank you for your patience and your participation in the first session of the webinar series on fermentation technology. The second session of the webinar series will start exactly at 3 o'clock today, and the YouTube link for the same will be shared to you via email as well as through the WhatsApp group created especially for this webinar. The feedback link has been posted in the chat box also. Kindly submit your feedback, and based upon the cumulative feedback submitted for all the 7 sessions, the certificate of participations will be issued to all the participants. See you all at 3 o'clock in the session 2. To be the resource person of session 2 is Dr. Samuel Jacob from SRM University, who will be talking on upstream processing of fermentation technology. The links will be shared. Kindly join us for the second session also. Thank you very much.
So we can end it. Feedback link. Yes, sir. Thank you, sir.